Welcome. Today, we're diving into drug release testing, specifically for topical dosage forms. I'm Dr. S.K. Singh, and together, we'll explore how medications applied to the skin release their active ingredients, a crucial step in ensuring safety and effectiveness. Let's get started. Let's start by understanding what drug release testing is all about. It's a way to measure how quickly and how much of a drug is released from a topical product, like a cream or gel, under controlled lab conditions. We use this testing to see how well a formulation performs, make sure every batch is consistent, and help guide development. It also plays a key role in spotting changes during manufacturing and supports regulatory updates after approval. In short, drug release testing is essential for quality control and assurance in semi-solid products. When we talk about GMP expectations, we're really focusing on a few key areas. First, the method we use must be validated. It has to be proven reliable. Then, controlling membrane integrity is crucial, ensuring the product performs as intended. Data traceability and integrity compliance are non-negotiable. Every step must be documented and trustworthy. Globally, agencies like the FDA, EMA, and ICH provide guidance to keep us aligned. For example, FDA's SUPACSS and product-specific guidances recommend IVRT for generic topicals. EMA and ICH offer quality and risk management frameworks, while USP chapters set standards for semi-solid and topical products. Together, these form the backbone of quality assurance in topical drug development. Let's take a closer look at the focus of two key in vitro tests. IVRT, or in vitro release testing, measures how the drug is released from its vehicle into a receptor medium. On the other hand, IVPT, or in vitro permeation testing, looks at how the drug moves through a biological membrane, like skin. These tests are part of a broader set of topical performance studies, each designed to assess different aspects, from drug release to skin penetration to clinical bioequivalence. They're essential tools for understanding how topical formulations perform. Let's dive into the principles behind in vitro release testing, or IVRT. This method helps us understand how a drug is released from its formulation, outside the body. It's a critical step, giving us early insight into how the medicine might perform. By carefully controlling conditions, we can measure release rates consistently, ensuring quality and predicting effectiveness. IVRT is a powerful tool for formulation development, stability studies, and regulatory approval. It's all about precision and reliability in the lab before we move forward. Let's take a closer look at the membranes we use in this process. Cellulose acetate, PTFE, nylon, and other synthetic membranes each play a role. The conditions are carefully controlled. Temperature stays around 32 degrees Celsius, with stirring between 300 and 600 RPM. We work with receptor volumes from 5 to 20 milliliters, and sampling happens anywhere from half an hour up to 24 hours. The typical setup includes a donor chamber, the membrane itself, a receptor chamber, plus a magnetic stirrer, sampling port, and a thermostatic water jacket to keep everything stable. Let's walk through the experimental procedure step by step. First, we prepare the receptor medium, the environment where our test will happen. Next, we install the membrane, which acts as a barrier in our setup. Then, we equilibrate everything at 32 degrees Celsius to mimic body conditions. After that, we apply the formulation, between 200 and 500 milligrams. Samples are collected at set intervals to track changes over time. These samples are analyzed using HPLC, UPLC, or UV spectroscopy. Finally, we calculate the cumulative drug release to understand the formulation's performance. Here, we see a table showing drug concentration measured over time, in micrograms per milliliter. At 1 hour, the concentration is 15. By 2 hours, it nearly doubles to 28. 4 hours in, it reaches 50, and at 6 hours, it's 68. This data forms the basis for calculating cumulative drug release, tracking how much of the drug has been released into the system over time. It's a clear example of how we quantify drug delivery in real terms. Let's talk about the Higuchi drug release model. It's one of the most common ways to describe how drugs release over time from a delivery system. This model helps us predict the rate at which a drug diffuses through a medium, often a solid matrix. It's simple, yet powerful, and widely used in pharmaceutical research to understand and control drug release profiles. Knowing this model gives us a solid foundation for designing effective therapies. Here, we see a table showing experimental data for release rates over time. The first column lists time in hours, starting at 1 hour and going up to 6. The second column shows the square root of time, which helps us analyze the release pattern. Finally, the third column gives the release amount in micrograms per square centimeter. This data lets us calculate how quickly the substance is released, giving insight into the underlying process. When we talk about method validation, there are a few key requirements we need to keep front and center. First, specificity, making sure nothing else, like excipients, interferes with our results. Then, linearity, our data should show a strong correlation, with an R-squared of at least 0.99. Accuracy means our recovery falls between 98 and 102%. Precision is tight, with a relative standard deviation under 5%. We also test robustness, checking how temperature, stirring rate, and membrane lot affect outcomes. Finally, system suitability ensures consistent performance across diffusion cells. When we look at what affects drug release, there are several key factors at play. Manufacturing steps like homogenization speed, mixing time, and temperature can all influence the final product. Then, packaging comes into the picture. Things like drug adsorption and stability changes matter a lot. On the drug side, solubility, particle size, and polymorphic form shape how the drug behaves. Finally, the vehicle itself, its viscosity, emulsion type, and surfactants, all contribute to the overall release profile. It's a complex balance. Now, let's talk about acceptance criteria and how we interpret data. Acceptance criteria are the specific conditions we set to know when a task or project is truly complete. They give us a clear target to aim for. Data interpretation, on the other hand, is about making sense of the numbers and information we collect. Together, these help us measure success and make informed decisions. Without them, we're just guessing. Let's look at some common regulatory deficiencies that often trip up submissions. First, method issues, like using a non-validated IVRT method, choosing the wrong membrane, or skipping sync condition checks. Then, data integrity problems, missing raw chromatograms, uncontrolled spreadsheets, or incomplete audit trails. Scientifically, poor reproducibility, high variability, and not enough diffusion cells can all raise red flags. These issues lead to serious consequences: FDA observations, deficiency letters, and ultimately delayed approvals. Let's wrap up with some key takeaways about IVRT, or in vitro release testing. It's a critical quality tool for topical formulations, helping us understand how drugs release from a product. The France diffusion cell remains the industry standard for this testing, with drug release usually following diffusion-controlled kinetics. Method validation is essential to meet regulatory expectations. IVRT supports everything from product development and quality control to SUPAC changes and generic approvals. When done right, scientifically sound, and GMP compliant, it ensures consistent performance, patient safety, and regulatory compliance. That brings us to the end. For any further support or questions, please don't hesitate to reach out. Thank you for your time and attention. It's been a pleasure sharing this with you.